Hello everyone, welcome to ITLS Academy. Today we are going to discuss the topic that is preparation and standardization of base and acid solution. So here what we are going to see, that we have to prepare our acid solution and we have to prepare a base solution, and along with it we have to do a standardization of this acid and base. Okay, so today we are going to discuss this. Now, introduction. See, many type of chemical analysis are made using a method in which a constitutes is treated with a solution of known strength to an indicator endpoint. Okay, we all know that there are several type of chemical analysis in which what should we do? We titrate the constitute with the solution whose strength is known, and we titrate it till the endpoint is reaches. Okay, now that type of solution are called what? Or called a standard solution. Okay, now we already discussed about the standard solution. Standard solution, we all know that standard solution are defined as whose concentration are known. Okay, suppose that there is a question in which it is written that we have to dissolve 5 gram of NaCl into a 100 ml of distilled water. Now in this 5 gram and 100 ml, we know the amount of solute and even we know the amount of solvent. So this is called what? This is called a standard solution. So in many type of analysis, chemical analysis, what should we do? We titrate the solution with the standard solution. Okay, and we titrate it till we get the end point. From the volume and concentration of standard solution used in the titration and the sample size, the concentration of the constituent in the sample can be calculated. Now how we calculate this constitute in the concentration of the constituent in the sample? With the help of volume and concentration of the standard solution which we use in the titration. Okay, and sample size. Okay, with the help of sample size, with the help of the sample size and volume and concentration of this standard solution, we find out or calculate the concentration of the constituent in the sample. Okay, the assay of titratable acidity is a volumetric method that uses a standard solution and most commonly the indicator phenolphthalein. Okay, so basically what should we do? We titrate acidity is a volumetric method in which what should we do? We use a standard solution, and the indicator which is commonly used is called a phenolphthalein, and phenolphthalein is used as a to detect the endpoint. Phenolphthalein is used to detect the end point. Okay, in a titration, standard solution of sodium hydroxide react with the organic acid present in a sample. Okay, suppose that there is a some process of titration we are performing in which we have a standard solution of sodium hydroxide, that is your NaOH. Now this NaOH is what they do? They react with the organic acid which are present into a sample, and the normality of the sodium hydroxide solution, the volume used and the volume of the test sample are used to calculate the titratable acidity. Now how we calculate the titratable acidity? With the help of the normality of the sodium hydroxide and volume, how much amount of the sodium hydroxide are used, and volume of the test sample. Now expressing it in terms of predominant acid present in a sample, a standard acid such as your potassium acid and phenolphthalein that can be used to determine the exact normality of the standard solution of sodium hydroxide which is used in a titration. So with the help of this standard acid like your potassium acid phthalate, what they do, they it is used to determine the exact normality of the standard sodium hydroxide solution which are used in a titration. Now come to the next slide. The phenolphthalein endpoint in the assay of titrable acidity is pH 8.8. Okay, so when we are performing this titration, then at that point the pH of the phenolphthalein indicator is what it is, 8.2, and where there is a significant color change from clear to pink. Okay, suppose that this is your clear color, then this color will be changed into a pink color. Just a minute. This is your pink color. Okay. Now when it changes clear to pink, then color of the solution obscure the pink endpoint, and the potentiometer method is commonly used. A pH meter is used to titrate such sample to a pH 8.2. Now this is all about the introduction of this experiment. Now the next one is your objective. So here the first one is what? First one is a preparation and sterilization of base and acid solution. Okay, so in this what we are going to see, we are going to see how we prepare sodium hydroxide solution and hydrochloric acid solution, that is your NaOH and HCl. Because this NaOH is what? It is a base, common base, and this HCl is what? HCl is a acid, common acid. Okay, now what we have to do, we have to prepare the sodium hydroxide and hydrochloric acid solution, and with the help of these solution we have to sterilize this solution. So this is the objective of today's experiment. We have to prepare the solution of one acid and one base, that is your sodium hydroxide, and hydrochloric acid, that is your HCl, and after that we have to sterilize this solution. Now principle, a standard acid can be used to determine the exact normality of the standard base and vice versa. Okay, that means that if we use a standard acid, then this standard acid are used, with the help of this standard acid, what should we do? We find out the exact normality of a standard base. Okay, now if we have a standard base, now this standard base are used to calculate the exact normality of standard acid. Okay, so standard base is used to determine the normality of standard acid, and standard acid are used to determine the normality of standard base. This is the principle of this experiment. Now the next one, now come to the next slide, that is your chemical use in this experiment. So here we use ascaride. Okay, the first chemical that we use is a ascaride. Ascaride case number 81133-20-2, and the hazard related to this chemical are what? They are corrosive in nature. Now the next chemical is what? It is ethanol, that is 6417-5. They are highly flammable. Okay, the next one is your hydrochloric acid, that is your HCl, 7647-01-0, that is corrosive. Phenolphthalein 77-09-8, irritant. Potassium acetate acid phthalate, that is 87724-7, that is irritant in nature. And the last chemical is your sodium hydroxide, that is your NaOH, case number 131073-2. They are what? They are corrosive in nature. So these are the list of the chemical and their hazard related to these chemical. So hazard means that when we have to use this chemical, we have to take a care that these type of hazard are related to these type of chemicals, so we have to take care. Now the next one is your reagent used in this experiment. The first reagent is your ascaride trap. So ascaride, for the preparation of this ascaride trap, what should we do? We simply take a ascaride and put it into a syringe, and this syringe are attached to a flask in which carbon dioxide free water are present. Okay, and that's how we prepare this ascaride trap. What should we do? Suppose that this is a syringe. Okay, now in syringe, what should we do? We put a ascaride. Okay, now this ascaride is attached to a flask. Suppose that this is your flask. Okay, now in this flask, what present here? It is present CO2 free water. Okay, this is how we prepare this ascaride. Now the next one is your how we prepare the carbon dioxide free water. For this we have to take a 1.5 liter. Sorry, we have to prepare a 1.5 liter of carbon dioxide free water for a person or a group. For this, what we have to do, we boil the deionized distilled water for a 15 minutes. Suppose that this is your beaker, and in beaker we have a deionized distilled water. Okay, so we have to boil it for a 15 minute, and this aluminum flask are what or of 2 liter. Okay, now after boiling, what should we do? We place a stopper and the flask with rubber stopper through a through which is inserted in a tube attached to a ascaride trap, and then we have to allow the water to cool down with the ascaride protection. This is how we prepare this carbon dioxide free water. And we required what? We required ethanol of 100 ml. So here if it is not written that how much percent of ethanol we required, so we required here a 99.9% of ethanol. Okay, we require a hydrochloric acid, and hydrochloric acid will be in a concentrated form. We required a phenolphthalein indicator solution of 1%, which means that we dissolve 1 gram of phenolphthalein phenolphthalein into 100 ml of distilled water. Then we get what? 1% of this phenolphthalein indicator. Here see it is written then, sorry, not water, we dissolve into 100 ml of ethanol. Okay, and see here it is written that dissolve 1.0 gram in 100 ml of ethanol and put it into a bottle with the eyedropper. Why we put it into a eye with the eyedropper? Because we have to require it in a drop form. Okay, now the next one is your potassium acetate acid phthalate, that is KHP. So for this we required 3 to 4 gram of dried in oven at 120 degrees Celsius for 2 hours. What should we do? We dry this for potassium acetate phthalate at 100 degrees Celsius for 2 hours. After cool down, we have to store it into a closed bottle and place it into a desiccator. Okay, until use. And the last reagent that we required is a sodium hydroxide palette. So this is a chemical that we require, that is a reagent that we required in our experiments. Now the next one is your hazard precaution and waste disposal. We all know that we have to follow all the no, all the laboratory procedure. Okay, and we have to wear a gloves, glasses, lab coat. Even we have to wear a mask, because this chemical which we are using are very hazardous. And along with it, we have also follow a protocol of our institution, and we have also follow a good laboratory practices which are outlined by a environmental health. Okay, and waste which is produced by performing this experiment are drained out by using a water rinse. Okay, now see what is written in a slide. Use a appropriate precaution in handling concentrated acid and base otherwise added to a normal laboratory safety procedure. Wear a gloves and safety glasses at all time. Waste likely may be put down the drain using a water rinse, but follow a good laboratory practices outlined by our environmental health and safety protocol at your institution. Okay, so now next one is your material required for this experiment. We required a beaker of 50 ml for the waste of NOH. Okay, when we fill this NOH into a burette, then there is some waste are produced. Then for this waste, we required a beaker of 50 ml. Another beaker is required of 100 ml, 25 ml or 50 ml. Okay, then we also required a 5 ml flask of 250 ml and 1 ml flask of 1 liter. Okay, so after that we required a funnel small to fit top of 25 or 50 ml of burette, glass stirring rod, glass storage bottle of 100 ml, graduated cylinder of 50 ml, graduated cylinder of 1 liter, graduated pipette of 1 ml. And we also required a graduated pipette of 100 ml, parafilm, pipette bulb or a pump, plastic bottle with the lid of 50 or 100 ml, and plastic bottle with the lid of 1 liter. Spatula, spirit bottle with the deionized distilled water, volumetric flask of 50 ml, volumetric flask of 100 ml, and weighing paper or a board, weighing board, and white piece of paper. So these are the material that is required for performing the experiment. Now the next one is equipment. We required three equipment for performing this experiment. One is your analytical balance for the weighing purpose. Okay, forced draft oven heated to 120 degrees Celsius and hot plate. Now the Now come to the procedure. First of all, we have to prepare a 25% weight by volume of NaOH solution. For this, what we have to do, we have to prepare a 50 ml of 25% of NaOH in deionized distilled water. To this, to do this, what we have to do, we have to take a weight of NaOH and put it into a 100 ml of beaker. Now, after that, we have to, suppose that um this is your NaOH. What should we do? We have to prepare a 25%. Then we take a 25 gram of NaOH and put it into a 100 ml of flask. Now we have to add a 40 ml of deionized distilled water and stir the NaOH palette with a glass stirring rod, continuous stirring until all the palette are dissolved. Then after that, we have to transfer the solution into a 50 ml of volumetric flask and dilute to a volume with a deionized distilled water. Then the remaining 60 ml of deionized distilled water will be added. When all the pallets of this NaOH uh, dissolved into the deionized water. Then after that we have to cool down the solution at a room temperature. Then we have before our final preparation. Then after that we have to store it into a plastic bottle and label appropriately. See, when you prepare a reagent into a lab, then one thing you have to take care, that is your labeling. Okay, suppose that when you are preparing this 25% of NaOH, so you have to write down that 25% of NaOH. Then after that, you have to prepare a 0.1 normal of HCl solution. For this, what should we require? We required a concentrated HCl 12.1 normal and deionized distilled water. Okay, now we have note down one thing: do not use mechanical pipette to prepare this, since the acid can easily get into the shaft of the pipetter and cause damage. Okay, here we are not using the mechanical pipette for for the preparation of this concentrated HCl, because this HCl are get into the shaft of the pipetter and cause damage. Now to prepare this solution, place a small amount of deionized distilled water into a 100 ml of volumetric flask. Suppose that this is your 100 ml of volumetric flask. Okay. Now what should you have to do? You have to add like 20 ml of deionized distilled water and appropriate amount of concentrated HCl. Then after diluting with the deionized distilled water, you have to mix it properly, and again you have to transfer it into a glass bottle, and after transferring into a glass bottle, you have to seal it and label appropriately. Now the next one is you have to prepare a 0.1 normal of NaOH. For this, we transfer 750 ml of CO2 free water into a 1 liter of plastic storage bottle. After that, we have to add a 12 ml of well mixed 25% of NaOH solution, which are prepared in a first step. See, what should we do? Uh, transfer 750 ml of CO2 free water into a 1 liter of plastic bottle. Okay, suppose that this is your plastic storage bottle. Here you have to transfer 750 ml of CO2 free water. Okay, now after that you have to add 12.0 or 12 ml of 25% of NaOH. Okay, then you have to mix this thoroughly, and this will give an approximately zero point 0.1 normal of solution. Now you have to fill it into a burette with the help of this funnel and discard the first volume of the burette and then refill the burette with NaOH solution. Okay, then you have to discard the first first volume of the burette. Then after that, again you have to fill it with the NaOH solution. Now you have to sterilize this zero point 0.1 normal of NaOH. For this, what should we do? Accurately weigh about 0.8 gram of dry potassium acetate phthalate in to each of 3 to 50 ml of aluminum flask. Suppose that these are three remedial flasks. Now, in each of the remedial flasks, you have to add 0.8 gram of potassium acetate phthalate. Okay. Now you have to record the exact weight. Suppose that maybe weight will be a 0.81, 0.82, sometime it is 0.9. So you have to record the exact weight, and after that you have to add a 50 ml of cool CO2 free water. Okay, now CO2 free water, 50 ml. Now seal the flask with the paraffin and swill gently until the sample is dissolved. Then we have to add a 3 drop of phenolphthalein indicator. After mixing it, we have to add a 3 drops of this phenolphthalein indicator and titrate against a white background with the help of this NaOH solution being sterilized. Now record the beginning and ending volume of the burette, and we have to do this process until we get a pink color which will persist for a 15 second after swirling. Now the color will fade with time. Record the total volume of the NaOH which is used in titrate in each sample, and data from this part will be used to calculate the mean normality of the diluted NaOH solution. So the data that we get from here, what should we do? We use it to calculate the mean normality of the diluted NaOH solution. Now the next one is we have to sterilize the 0.1 normal of HCl solution. For this, again we have devised the same scheme, determine the exact normality, and we use 0.1 normal of HCl solution which are already prepared in a step 2. Now you have to remember that you have sterilized NaOH to use, do analysis at least duplicate and record the volume which is used. Now the next one is your data and calculation. Okay, so here using the weight of KHP and the volume of NaOH which is titrated in the above section. Step 4, calculate the normality of the diluted NaOH solution and determined by each titration, and after that you have to calculate the mean normality, and mean normality are calculated by the molecular we have to calculate the mean normality, and the molecular weight of this potassium acetate acid phthalate is what 204.228 gram, and the range of triplicate determined for normality should be less than 0.2% with good technique. Okay, now that so this is the table for the experiment. Here what should we do? We perform it into a triplicate, like we take a 3 samples. Okay, so here weight of the KHP, this is your burette start, burette end, and with the help of this, we find out the volume of NaOH which is used and normality of NaOH. Now, sample calculation. We all know that suppose that we get a weight of KHP is 0.8115 gram. Now, we all know that the molecular weight of KHP is what 204.228 gram per mole. Now, volume of 0.10 normal of NaOH that is used in titration will be 39 ml. Okay, and mole of KHP is what we divide this weight of KHP with the molecular weight, and mole of KHP of NaOH is equal to here. So this is the normality of the NaOH. Now with the volume of HCl and NaOH which is used in the above section, that is step 5, we have to calculate the exact normality of the HCl solution, and it is determined by each titration, and then we have to calculate the mean normality. Here the volume of NaCl that is used and volume of NaOH that is used, and here it is a normality of the HCl, and with the help of this we calculate the mean of this. So this is all about the today's lecture. I hope you will understand all the things. If you have any doubt, just send your doubt to the in the comment box and ask freely. For a further information, you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also send your query to the helpline number that are already mentioned in your account, and you can visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe. If you have any query, just let us know in a comment box.